We're going to take a look at chemical messengers and their classification by solubility, so whether they're hydrophilic or hydrophobic. Let's take a look at the hydrophilic messengers first. So remember that things that are hydrophilic cannot diffuse through the cell membrane. So remember the middle part of the membrane where the fatty acid tails are on the phospholipids? Remember that's hydrophobic in the middle, and that's going to stop any of the hydrophilic messengers from diffusing across, so they will not be able to diffuse through the membrane. When the hydrophilic messengers are made, they can be stored in vesicles, which are membrane-bound sacs, and then they can be released later. How they're going to be released from the cell is by exocytosis. So the vesicle with the hydrophilic messenger will come up and fuse with the cell membrane and then release the contents. And if the hydrophilic messenger is a hormone, it mixes very well with the water in the blood. So there's no carrier protein that it needs to be attached to while it's in the blood. This is just a picture showing an endocrine gland secreting a hydrophilic messenger by exocytosis. So maybe this is insulin hormone, for example, a protein. And then when it goes into the bloodstream, it just dissolves very easily in the blood, and so it doesn't need to be attached to a carrier protein at all. When the hydrophilic messenger gets to its target cell, it's going to bind to its receptor, which is going to be located on the cell surface. So these receptors will be in the cell membrane of the target cell, because you wouldn't want to have any receptors that are on the inside of the cell, because the hydrophilic messenger wouldn't be able to get inside. So these receptors will be on the cell surface, and that's where the hydrophilic messenger will bind to its receptor. And then the target cell response, once the messenger binds to its receptor, will be either to turn enzymes on or off, or to open or close ion channels. And both of those are going to be fast in onset. So especially if you think about ion channels, opening and closing that gate happens very quickly. And the target cell response will be short-lived. For example, the ion channels, when they open, the gate's going to close very quickly after it opens. And this is just a picture just showing the hydrophilic messengers binding to the receptors, which are on the cell surface. So they're embedded in the plasma membrane of the target cell. And then very important to know the three types of hydrophilic messengers. So you want to memorize that the amino acids are hydrophilic, and the amines, which are made from amino acids, are also hydrophilic, with the exception of thyroid hormones. Thyroid hormones are actually hydrophobic. And then we also have messengers that are made out of proteins or peptides. Those are also hydrophilic. And remember that proteins are made out of amino acids that are joined together, so it's not surprising that the peptides and proteins would be hydrophilic as well. So as long as you can remember that the amino acids are hydrophilic, and you know that amines and the proteins are made from amino acids, then you know that all three of those will be hydrophilic. Hydrophobic messengers are really different because they are able to diffuse right through the cell membrane. So they can cross the membrane easily. As soon as the hydrophobic messengers are made, they are immediately released from that secretory cell. So they're not able to be stored in vesicles, because remember, a vesicle is made out of a membrane sac, and these hydrophobic messengers just diffuse right through membranes, so you can't store it in a vesicle. So as soon as they're made, they just get released. And how they're released from the cell is they're just going to be released by diffusion. So they just diffuse right out of the cell membrane of that secretory cell and then go to wherever they're headed to. If the hydrophobic messenger is a hormone, it's going to be transported in the blood attached to a carrier protein. Because it's hydrophobic, it's not going to mix very well with the water in the blood. So we're going to attach it to a hydrophilic carrier protein, which will make the hydrophobic hormone mix better with the blood. This picture shows an endocrine cell secreting a hydrophobic hormone. So maybe this is cortisol or estrogen or testosterone. And you can see that as soon as the hydrophobic hormone is made, it's going to be released by diffusion right through the plasma membrane. And then when it enters into the blood, because it's hydrophobic and doesn't mix well with the water in the blood, it's going to be attached to a carrier protein. And remember, proteins are hydrophilic, and that'll make the hydrophobic hormone mix better with the water in the blood. The hydrophobic hormone does not bind to the carrier protein permanently. It will bind and then fall off. And when the hormone falls off the carrier protein, then it's free and can then diffuse out through the capillary wall and then go to its target cell. But while it's bound to the carrier protein, it's trapped in the blood. So as far as where the receptors are located on the target cells, they're going to be inside the cell, so either in the nucleus or in the cytoplasm. So they will be inside the cell, in the cytoplasm, or the nucleus. And when the hydrophobic messenger binds to its receptor inside the cell, the target cell response is to turn genes on and off. So in order to turn genes on and off, that's going to take some time. So the target cell response will be slow in onset. It's going to take a while to occur. And then the target cell response is going to be long-lasting, because if you're turning these genes on and off, that's going to affect which proteins you make. And proteins are valuable, and usually they're going to stick around for a while, so the response will be long-lasting. And then it's important to remember which chemical messengers are hydrophobic. So we have the steroids, eicosanoids, and then we have that exception with the amines, because remember, the amines are normally hydrophilic except for the thyroid hormones, so that's hydrophobic. And this is just a picture just showing the hydrophobic messengers binding to the receptor, which is inside the cell. So the reason the receptors are inside the cell is because the hydrophobic messenger can just diffuse right through the plasma membrane of this target cell. And so it'll bind to a receptor located either in the cytoplasm or in the nucleus. And here's just an example of a hydrophobic messenger. So let's say that we have the hormone testosterone here, and it's going to diffuse right into the skeletal muscle cell. And notice that the testosterone receptor is inside the cell, and this one happens to be in the cytoplasm. And if they're in the cytoplasm, once it binds to the testosterone, the receptor in testosterone will then move into the nucleus and actually bind to the DNA and turn genes on and off. And in the case of testosterone in skeletal muscle, it will turn on genes that tell the cell to make the contractile proteins, the actin and myosin. And so that's going to take some time to turn these genes on, transcribe them, make the messenger RNA, and then translate them with the ribosomes and make the new contractile proteins. So it'll be a slower onset response, but it's going to be long lasting because those new contractile proteins that have been made are going to be hanging around for a while. And this is why men have bigger muscles than women do, because they have more testosterone that they make, and that tells the skeletal muscles to make more of these contractile proteins. And so their muscles are more full of these contractile proteins, and so they're bigger and stronger than women. And last thing to talk about is the half-life of hormones. Half-life is just defined as the time it takes for half the hormone in the blood to be degraded or broken down and gotten rid of. So hydrophilic hormones, they are going to have a short half-life. And that's because they are not bound to the carrier proteins when they're in the blood. And so they're not protected, and so it's pretty easy to break them down and get rid of them. The hydrophobic hormones will have a long half-life, and that's because they are attached to the carrier proteins in the blood. So when they're attached to the carrier proteins, they can't get broken down or get out to their target cell, and so they're going to hang around longer, so they will have a longer half-life. So here's just an example showing you some of the hydrophobic hormones. We have thyroxine, or T4, which we'll learn about in Chapter 6. And then we also have some steroid hormones. We have cortisol and testosterone. And notice that most of these are going to be very highly bound to the carrier proteins while they're in the blood. And you can see the half-life is pretty long. So for example, for thyroxine, that thyroid hormone, it takes 6 days to get rid of half of the thyroid hormone that's in the blood. So that's a really long half-life. And then these protein hormones that are on the bottom here, remember proteins are made of amino acids which are hydrophilic. So these are not bound to the carrier proteins in the blood, and so they're not protected, and so they have a really short half-life. So for example, insulin has a half-life of 8 minutes. So it only takes 8 minutes to get rid of half the insulin in the blood. And this is just a picture of these hydrophobic hormones and why they have such a long half-life. So here's our endocrine cell that is going to be secreting our hydrophobic hormone, and it will just diffuse out of that cell and enter into the blood, and it will bind very quickly to our carrier proteins in the blood, and there's a picture here to remind you of that. And while it's bound to the carrier protein, it can't get to its target cell and be degraded there. It also is protected from enzymes in the blood and also the liver, and so it's safe and it can't be broken down. So hormones that are bound to these carrier proteins are going to have a much longer half-life because they're protected. But remember that we said that the hormone is not bound to the carrier protein permanently, so it will bind and then fall off. And when it falls off, it can diffuse out of the blood and go to the target cell and bind to its receptor there. And then it can get destroyed at the target cell, or if the hormone is still traveling around the blood and it's free, it could be degraded by enzymes in the blood or also by enzymes in the liver as well.